hypothesized that resveratrol increases the inflow from mitochondrial content in mice by activation of AMPK, which in turn increases production of PGC1 alpha. This activation of AMPK by resveratrol does not occur in humans, meaning that they do not see the same increases in mitochondrial content as mice.
Our match for this problem is mitochondrial content, our inflow is protein synthesis, and our outflow is protein breakdown. However, for this problem, we'll be focusing primarily on inflow and won't be addressing the outflow in any more detail. AMPK increases protein synthesis via phosphorylation of PGC1 alpha. This occurs when exercise increases the breakdown of ATP to ADP, which concurrently increases concentrations of AMP. AMP directly activates AMPK, which both directly phosphorylates and activates PGC1 alpha, and phosphorylates NAMPT. NAMPT oxidizes NADH to NAD+, which activates SIRT1. SIRT1 then deacylates PGC1 alpha, also activating it. Activated PGC1 alpha is translocated to the nucleus, where it acts as a transcription cofactor for mitochondrial gene transcription. This transcription produces mitochondrial gene mRNA, which is then translated in the rough endoplasmic reticulum and results in mitochondrial protein synthesis. Resveratrol directly activates AMPK, which via both pathways, direct phosphorylation of PGC1 alpha and indirectly through activating SIRT1, and therefore deacylating PGC1 alpha, contributes to increased protein synthesis and therefore increases in mitochondrial protein content.
To test our hypothesis, we examined the evidence in a journal article called AMP-activated protein kinase deficient mice are resistant to the metabolic effects of resveratrol. The subjects of the study were mice deficient in a catalytic subunit of AMPK and wild-type mice. They were fed high-fat diets or high-fat diets with resveratrol for 13 weeks. This figure shows mRNA levels of PGC1 alpha in the wild-type and AMPK knockout fibroblasts after resveratrol treatment. Resveratrol induced the expression of PGC1 alpha in wild-type but not knockout fibroblasts. The following figure shows transcription levels of skeletal muscle genes as induced by resveratrol. Transcription of PGC1 alpha increased in wild-type but not AMPK knockout mice after exercise. Resveratrol increased mitochondrial DNA in wild-type but not AMPK knockout mice after exercise. This supports our hypothesis that resveratrol increases mitochondrial protein synthesis in mice. This figure shows cytochrome C and SIRT1 levels in wild-type and AMPK knockout fibroblasts after resveratrol treatment. Cellular cytochrome C, which is a marker of mitochondrial content, and AMPK increased in wild-type but not AMPK knockout fibroblasts. There was no change in SIRT1. This supports our hypothesis that resveratrol activates mitochondrial biogenesis in mice and shows that this process likely occurs via the AMPK pathway and not via direct activation of SIRT1. Activity of AMPK as indicated by ACC phosphorylation and phosphorylation of AMPK increases with resveratrol treatment in wild-type mice and wild-type fibroblasts. There was no difference in SIRT1 between resveratrol and control conditions. This supports our proposed mechanism of mitochondrial biogenesis through the AMPK pathway in mice. Acetylation levels of PGC1 alpha in skeletal muscle is increased by resveratrol treatment in wild-type but not AMPK knockout mice. This supports our hypothesis that resveratrol activates mitochondrial biogenesis in mice via the AMPK pathway. The NAD-NADH ratio increased in wild-type mice treated with resveratrol after exercise, but not AMPK knockout mice. This supports our proposed mechanism of AMPK activating mitochondrial biogenesis via PGC1 alpha indirectly via SIRT1 in mice. These results support our hypothesis that resveratrol acts directly on AMPK in mice to induce mitochondrial biogenesis. To test our hypothesis, we looked at a study published in 2012 called Acute Exposure to Resveratrol Inhibits AMPK Activity in Human Skeletal Muscle Cells. The figure shown partially supports our hypothesis. RSV at 1 micromoles per liter inhibited AMPK activity by 50% and AMPK alpha 2 activity by 35%. Additionally, resveratrol at 100 micromoles per liter totally blocked the activity of both AMPK isoform complexes in the in vitro assays. We correctly hypothesized that resveratrol does not activate AMPK in humans and thus would not increase protein synthesis and inflow from mitochondrial content.